Thank you. Thank you, Bina. It's been a pleasure to work with you guys, and I'll always speak very easily so everybody can follow. And then if we need like more specific details, I can give you after the presentation here. All right, so I think so the idea of the project was to start modeling 8P using human brain organoids. And I think at that stage, I mean, you all know that we can use stem cells to create these brain organoid models that mimics neurodevelopment. And being 8P a genetic condition, we can capture the genomic structures of 8P in a pluripotent state, and from that pluripotent state, we can guide or cue the cells to become a neurotissue. And that has several advantages. We can study these neurotissues to figure out how the disease happens, and also to find therapeutic opportunities. So it's a very powerful model. This is not new in science, but it's something that over the years pick up lots of momentum. In the past, we used to rely on either postmortem tissues or animal models, and all of that have specific limitations. So brain organoids kind of a bridge a gap here in the knowledge, and that's the tool that I use in my lab. So before I continue, let me just highlight here the goals of the proposal that we have. The idea was to start with a trio, one proband and the two biological parents, reprogram the cells, make iPSCs. Actually, I mean, the cells were reprogrammed to me, so I just expand the cells and confirm pluripotency, making sure that this has no mycoplasm, that the chromosomes are contain the genetic variation that we're studying, things like that. And then, I mean, the idea once we have robust iPSCs, you start the differentiation into brain organoids, and then keep that differentiation going for several months. And throughout my talk, you will understand why I choose specific time points, meaning from 1 to 6 months. And the idea is that during that period, we can capture cell proliferation, cell differentiation, cell migration, and the beginning of network formation. We're going to do that using, I mean, as I said, the 3 iPSCs. And over time, we're going to collect samples from those batches of organoids to perform some global gene expression, basically bulk RNA-seq. But we're also going to save for single-cell RNA-seq. That would allow us to really understand what's going on in each of the cell types that we have in these organoids. And because that's a very dynamic process, we're going to see how these cells evolve over time, carrying or not the genetic alteration. So this is for my colleague scientists. I mean, we see this as a preliminary data. So we're building some arguments here using like one trio. Of course, we would like to see that with more families. We like to reproduce that with more families to understand what is common among all the families, and also what's different, what is specific to specific subjects. To do that, we have to dramatically expand the scope of this project and involve multiple families. But nonetheless, we have to start somewhere, and I think starting with a single family is what we decided that we need to do in order to generate preliminary data, so we can apply for larger grants and afford doing that with multiple families. And usually that's what I do with other foundations as well. So going back to why we use brain organoids, because these are all genetic conditions that started in uterus. So that very hard for us scientists to access experimentally how the human brain is formed inside the uterus. So we thought that by recreating the nervous system in the lab, we could analyze the different stages, how the brain is formed, and then, I mean, understand the consequences later in life. So considering the heavy neurological consequences that 8P has, I mean, we thought that this would be like a good approach. And as I mentioned, this is not a new technology. This was developed by a colleague of mine called Yoshiki Sasai back in Japan in 2008. that was exactly when I was starting my lab here at UCSD. And I thought, well, when I saw his first manuscript in describing the generation of these brain structures using iPSCs, I thought, wow, that's amazing. That's exactly what I want to do. And over the years, not only my lab, but several other labs have taken his protocol and developing it further, making it more robust, and also applying for different conditions. So we usually divide the brain organoid into two types. One is unguided differentiation, when you don't kill the cells to a specific brain region. So you actually get lots of brain regions in a single unit. So that has the advantage of the diversification of the brain regions. So you might have hippocampus, cortex, striatum, all in one, but they are very disorganized. And each one of these organoids will generate a very different structure. So it's hard to compare one to one. So for disease modeling, most of us end up using what we call guided differentiation. This is when you add specific factors in the culture condition to guide the cells to specific brain regions. So at least you define the brain region of interest. In my lab, I focus on the cortex, because the cortex is the center of human cognition and is the region that is mostly affected in these neurodevelopmental conditions. More recently, people are finding different protocols for different brain regions, and you can combine them, create what we call now assembloids. So you start creating or mimicking how the different brain regions form like small circuitry in a dish. It's very far from what really happens in the brain, but allows you to start modeling those interactions between the different brain regions. But for the simplicity, for the sake of simplicity here, we are focusing on the cortex. So let's just see what happens in the cortex and forget for a moment other brain regions. And before I continue, I always like to tell, especially for the families, the limitations of this approach, because this is not perfect. And most of the time, you're going to read in the media that the group create like mini brains in a dish and give you the impression that you actually have an entire brain in a petri dish, which is not true. So I'll show you what we actually have. And there are intrinsic limitations of this technology. For example, most of the cell types or neurons that we get are quite immature because they represent early stages of neurodevelopment. These organoids are not vascularized, so that creates a problem for them to continue to grow. They have a limitation on size. Not all cell types are represented. We do see a diversification, but not with all cell types that we see in the brain. We don't even know if the way we grow with cells are ideal or not. So there is room for optimization here as well. So with all these limitations, I mean, we still think that this is a useful tool for what we are doing, especially in the absence of a robust animal model where we can experiment on. So over the years, my lab has optimized the protocol for cortical organoids, and this was a protocol that we did with two postdocs that really took the time to optimize every single stage in these organoids. And right now, this is our formula. We basically start with single cells, and then we neuralize them, or we add the factors that induces them to become brain cells very early on, very transient. And then we took it off, these factors, and we add factors to stimulate the proliferation of these neuroprogenitor cells. These neuroprogenitor cells start to replicate and migrate and start to form the cortical plate. We then remove these growth factors and let the cells mature by themselves. So by doing that, those neurons start to send processes to each other to form connections or synapses. And over time, they mature and start talking to each other using electrical signal. And I'll show you in a moment how this happens. So you see that we start with single cells, and we end up with 5 million cells in each one of these spheres here. So each one of these balls have 5 million cells. That's the equivalent of a bee brain. And you can imagine how complex a bee brain is already. It is. So a question that I get often is what kind of cells you have inside these organoids. Before I tell you that, this is a postdoc in the lab. and she's handling a plate with a batch of organoids. Each white dot in there is one organoid with 5 million cells. So you can see that in a single batch, I generate enough organoids for different experiments. So I can do immunostainings, I can do RNA, I can collect for DNA, things like that. So it's very robust protocol. And the way it happens is by forming the cortex from inside out. This is exactly what happens in nature. So this is a two-dimensional representation of the organoids, where the center of the organoids is on top. And you can see the migrating neuroprogenitor cells. So they migrate from the core of the organoid to the outside. As they move along, you probably notice that they use some of these cables. These cables are what we call radioglia cells that guides the progenitor cells from the inside to the outside, where they're going to form the cortical plate. So after a while, in a cross section of these organoids, what you have is a ventricular zone, sometimes multiple ventricular zones. They are surrounded by neuroprogenitor cells. The radioglia cells sending process are here in white, and you see this beautiful formation of the cortical plate. If I show this image to a neuroanatomy, he probably cannot distinguish if this was lab-made or human-made. So it's very similar in anatomy, at least in these early stages, to how the human brains form. So that gives us some orientation of the cells and allow us for some kind of structure in these organoids that you cannot achieve, for example, if you have only a two-dimensional structure. So you require the 3D structures to actually see this nice formation of the cortex, for example. So here you go back to the cell types that we have in the organoids. And as I said, first stages are single cells, pluripotent cells, and the stages are 5 million cells in there. This is a snapshot at 4 months old. You have a pool of neuroprogenitor cells. These neuroprogenitor cells are making mostly glutamatergic neurons. At 4 months, we start to see a division between the upper and the lower cortical plate. So these are two of the major structures in the cortex where the neurons will diversify and help to structure all the circuitry in the cortex. Some of these progenitor cells are becoming gliogenic. So they are no longer producing neurons, but they are also producing glia cells, in this case, astrocytes. So astrocytes peak at 4 months, and then they start to increase the percentage inside the organoid. They will become 1-to-1 to neurons by 6 months. So half of these 5 million cells at the end are astrocytes. That's a time point that I pick in purpose, because at 4 months, with the astrocytes, we start shaping the synapses. That's when the electrical activity or the synapses almost exponentially start to grow inside the organoids to give rise to the first circuitry that we have in there. You also start to see a small, very discrete population of GABAergic neurons. So these are your inhibitory neurons. They are not inhibitory yet. They will become fully inhibitory a little bit later on. But at that stage, they're already showing markers of inhibitory neurons such as GABA. So they start to produce some of these receptors or start to release some of the GABA neurotransmitter inside the organoids. So we next ask if these organoids can actually form these neural connections or networks. And that was important because having the structures allows you to look for damages on the structural side. But sometimes, I mean, the problems are in the network, how the neurons connect to each other. So having organoids without networks is like having a pancreas without insulin. So there is a limitation in the functional assays that you can do here. So we put our organoids to the test by plating them on top of multi-electrode arrays. So these are electrodes that will touch the surface of the organoid in a still two-dimensional representation. So it's very similar to the EEGs that you place the electrodes through your skull and you capture the electrical activity from neurons that are nearby. But the beauty of this strategy is that you can do it longitudinally. You can measure the electrical activity over time and see how these networks evolve. That was never really done for organoids before, because most of the protocols didn't have both excitation and inhibition. Here is just like some pharmacology confirming that if you inhibit the glutamatergic neurons, the overall activity of the network goes down. If you inhibit the GABAergic neurons, the overall activity goes up. And you can also measure the release of these neurotransmitters using mass spec inside the organoids. So we see that they start producing GABA by the time the GABAergic neurons start to mature. So these all make sense, and we fully characterize this protocol. That's why we are proposing to use it. And this is the overall electrical activity over time. Our organoid is here in this red curve here. You can see that by 25 weeks, more or less around 4-month-old, you start to see a peak of electrical activity. And that's when we see a dramatic number of synaptogenesis going on inside these organoids. By 40 weeks or 9 months of age, these organoids are firing to something similar to a primate brain. So they reach these 20 hertz. This was never actually achieved in tissue culture before. So this is the first time that we see that level of activity. I plotted here some of the previous experiences from people using 2D cultures or 3D cultures. So the optimization of the protocol really pays off in terms of measuring the electrical activity. All right. One question that I always get, how similar are the organoids to the human brain? So we decided to do that by using the brain waves that we can collect from these organoids. So similar to the EEG, these brain waves are have different frequencies, and they change upon the different tasks that some humans are doing. And by the way, this is what cognitive neuroscientists have been doing for several years. So they are able to correlate all these different neural oscillations, alpha, beta, gamma, with human behavior. So we know, for example, that gamma oscillations are affected in schizophrenia. We know that if you have a seizure, some of these oscillations will change in frequency in amplitude. So we know that. We know how to correlate oscillations with human behavior. What has been missing is how to bridge the basic biology that we can get from the tissue culture to the neural oscillations in human behavior. And that's why we decided to do that. So long story short, what we show is that by 2 months, we don't have oscillations. We start seeing the oscillations exactly at 4 months, which coincides with the number of synaptogenesis being increased and the number of astrocytes inside this culture. The oscillations become quite rhythmically synchronized by 6 months and very complex after that, due specifically to the functionality of the inhibitory neurons. So if you block the inhibitory neurons, you never reach the level of complexity. And we can use like a machine learning algorithm to really matches that with human neurodevelopment. So here we took EEGs from preterm babies between 25 and 38 weeks and post-conception. And then we trained the machine to recognize the age of the subject based on the EEG. And then we feed the machine with the data coming from the organoid. So this is the multi-electrode data. So the machine has no idea if it's looking to the human data or the organoid data. And we ask, very unbiased, can the machine tell the age of the organoid using the human brain as a standard? And the data was simply amazing because the machine gets really confused. You cannot distinguish between a human brain or a brain organoid. So this is one of these eureka moments in the lab, showing here the predicted age versus the actual age. The perfect prediction is in the dotted line. The human data is in black, and the organoid data here is in blue. So each one of these blue dots is one organoid. So you can see that although it's more variable, so it's definitely not perfect, but it follows the same trajectory as the human brain. So it's maturing, it's creating electrical activity as the human brain does during development. Before 25 weeks, the prediction is very poor. And this is because we had no way to train the machine with human data on the first 25 weeks of this gestation, simply because that data do not exist. It's still a black box what happens to the human brain in the first 25 weeks of gestation. So we hope that the organoid will actually teach us something about that, but right now we really cannot compare. It would be impossible to compare. All right, why I'm using this method here, I'll give you like a quick example on how we move from causation to treatment. You probably remember the 2015-2016 outbreak of Zika virus in Brazil. The Zika virus was one of the candidates that could be causing these microcephalic phenotypes that we see in these babies. So by exposing the Zika virus to the organoids, we clearly showed that the Zika virus was able to kill these neuroprogenitor cells, avoid the migration of these progenitor cells, and reducing the cortical thickness, exactly what happens in the macrocephalic babies in Brazil. And that was a specific Brazilian variant that can do that. We also took advantage of this model to repurpose retroviral drugs, and we were able to find sofosbuvir as a drug that can be that can block the vertical transmission, meaning that if you have like a pregnant female that is infected by Zika virus, you can treat with sofosbuvir and the baby is born without the virus. So we were able to move from causation to treatment in just 2 years. So it shows how promising is the technology once you have it. If we didn't have the brain organoids, we would rely on animal models and the data would be inconclusive. We actually did that in parallel. So that's why we do it. All right, let me move on to what we have done so far for the project 8P. So as I mentioned, I mean, we expand these iPSCs and we generate these brain organoids, and we've been collecting this brain organoid for the past 5 months. So unfortunately, they got contaminated when we were able to, when we were about to collect for the 6-month time point. So this happens. These are contaminations that sometimes happens in tissue culture. It's hard to avoid, especially for these long-term cultures. So we have to keep an eye on that. So we are already starting like a new batch, so we can complete the study. The data from 1 month, 2 months, 3, 4, and 5 are collected, and most of the time, as I mentioned, we are collecting for single-cell RNA-seq. We are collecting for bulk RNA-seq. We are doing some immunostainings to see specific cell populations inside these organoids. We also look for cell death, proliferation, and any potential alteration that we already can detect at these very early stages. In 1 or 2 months, this is our trio here. You see the organoids forming 3 days, 15 days, 30 days, and 6 days. So there are some variability in the beginning, but they tend to become more homogeneous. These small ones that you see here will eventually die and be removed from the cultures, and you end up with these larger organoids. So just by looking at those images, you can tell that, I mean, even with the 8P mutation, these organoids were able to form and progress through development, which is reassuring because, I mean, we know that we have individuals with 8P with brains, so it would be a big surprise if they cannot make that. But you never know. We also measure the diameter of these organoids, day 3, day 15, day 30, and day 60. I wouldn't pay too much attention between the 3 and 30 days. But at day 60, it becomes clear that the proband it is probably smaller, or the diameter it is smaller than the two parents. There is some tendency for one of the parents to show a small decrease as well. That could be normal variation or some susceptibility, some genetic susceptibility on cell migration or proliferation. And by the way, that was the next question, why we have these smaller size organoids. So we look for proliferation. This is a simple assay. It's an imaging assay that we quantify the number of proliferative cells. And we don't see like major differences in these organoids. They're all about 85% cells that are proliferating. These are very early days, so most of the cells in there are neuroprogenitor cells, so they are cycling and they are producing neurons, as I mentioned. So we don't see like major differences here. Then we look into apoptosis. This is 60-day apoptosis data from the organoids, and we start to see some early apoptotic here on the proband or more apoptosis compared to the other ones. So you can see that doesn't seem like a huge or significant at that stage. And one reason that's when sometimes we see something like that is that we may be discarding the dead cells from the culture. So in the next time points, we actually collected the cells that were dead in the cultures as well, so we can have that measurement and confirm if there is more cell death or not. So this is just food for thought. All right, so I think Ethan asking me to end it with what is the translational relevance of this work? I mean, how can this work actually help on the clinical side? So this is really exploratory basic science, but there are some information that we can take from this data. The first thing is if there is a potential driver gene in this CNV that we might want to anchor our further strategies to develop perhaps an ASO strategy or a gene therapy strategy using that driver gene. Sometimes large CNVs, they have multiple genes, but just one is really the one that pushes forward the pathological condition. So if that's the case, we have lots of good tools to manipulate a single gene, but we are not good on manipulating multiple genes at the same time. So there is one potential, if there is one potential candidate, a single gene that might be driving that, we might be able to pick it up. And that will open other ideas. So these models also allows you to find molecular cellular phenotypes readouts for screening. If you want to find, for example, drugs to treat something, you need to show that the drugs are doing something. So that's why you need models, and that's why these models need to show some changes. So you need to have some changes to show that your treatment can actually revert or rescue or ameliorate the problem. So these organoids comes in handy by doing this type of screening for drugs or other things. Another thing that happens is sometimes you don't have a driver gene, but you have a pathway, a specific pathway, molecular pathway in the cells that might explain the problems, might explain the phenotypes, and it's passive for a drug treatment. So you don't need a single gene to do that. That can be done using drugs. Even available drugs might be able to repurpose that for those things. And the fact that we are doing single cells will allow us to see what kind of cells are affected the most, if not all. I mean, if there is a specific cell type that is more problematic in this condition than the others. So that might help us to narrow down our strategic pathways to a specific cell type. So it might be beneficial in the future. So these are some of the advantages that by completing this study, we might have, even though it's preliminary, but it might help in all of those. So we've done that for the 16p copy number variation. 16p comes in two flavors, a deletion and duplication. And this is a recent paper. It's a collaboration between my lab and Lilia's Yakosheva lab here at UCSD. We team up to model the 16p duplications and deletions. Long story short, what we find out is that no matter if it's a deletion or a duplication, there is one pathway that is the active ROA pathway that seems upregulated in both duplications and deletions. So that was a good indication that perhaps we can use that pathway to ameliorate the condition. So we used that And we tested with a drug called rosine that inhibit this active ROA pathway to see if we can rescue or restore the problem in those brain organoids. The major defect that we saw is the migration of these neuroprogenitor cells, all these dots here. These are progenitor cells that are migrating. You can see that both in the deletions as well as in duplication. The proliferation is not as strong or robust as we see in the controls. But after you add rosine, you see that we are able to restore the migration of these cells. So we are now exploring rosine as a potential clinical treatment. People was never treated with rosine before, so we don't know the toxicity, we don't know the time frame or the dose, the exposure. So there's lots of work to do, but at least we have a candidate that might work for both types of CNV in 16p. So this would be like a fantastic outcome of this research if we can do that. All right, I'll stop here. Thanks members of my lab who is doing this work, and funding agents and IMH and CERN mostly. And I'll be happy to take any comments and questions. Thank you.
Thank you so much. All right. You can use the chat or just unmute yourself, turn on your camera if you want to ask a question. Don't be shy. I have a question, but I want to defer to audience first. Looks like Nicoletta, you've got your hand raised. Please go for it.
Hello, thank you very much for your talk. That was really great. I have a few questions actually. We didn't really work ourselves with brain organoids, although it was always on the list of things to do. But in terms of general, as a general question for the technique, so when you get to your 5 million cells, what happens then? Are they renewing, or they are sitting there getting mature and perhaps older?
Yeah, by that time, we get 5 million cells when they're about 4 months, and the cells basically start reducing the proliferation, and the neurons just continue to mature. Because the organoids are not vascularized, the center of the organoids stop receiving the nutrients, because this mostly comes by diffusion. So with 5 million cells, you already have a deficiency inside of the core of the organoids. When these organoids are about 6 to 8 months, the center of the organoids will eventually become necrotic. But nonetheless, all the surrounding of the organoids remains alive. We can actually keep them alive for several years.
All right, great. And for the 8p organoids, are the cells the same size or similar size, or is it likely that they would be maybe smaller because there is not much proliferation difference and the apoptosis may be slightly different, but not so much? Did you look at the size of the cells?
That's a great question. We have not looked at that, but our immune stainings are coming, so we're going to have like a sense if the size is different. Yeah.
And on the MEAs, when you do the time course, are you using the same organoids? You keep them growing on the MEA, or do you move them on the MEAs before experiments, or how does it work?
No, once they are attached, they stay there. So we just take like some organoids from our pool, and then we plate on the MEA, and then we keep them on the MEA.
And do they move around in weeks?
No, they stick to the organoids and stay there forever.
All right, thank you.
Welcome. Yeah.
Is there a precedent for the cells to be smaller in previous studies you've looked at, or because I guess assessing the cell size, you'd have to, even if it's a small difference in each cell, I guess integrated across the whole mass of the tissue. But I guess is there a precedent for that, or is that? And that's a great question. I'm wondering if there's a
if you look at Rett syndrome neurons have the diameter of the neurons that is about 10% lower than the controls. And if you think about the volume, then we are talking about a big volume. So that creates the macrocephalic phenotype in Rett syndrome. So yeah, there are conditions where we see that. Yeah.
Perfect. Thank you. Faye.
Hey, that was an absolutely brilliant talk. Thank you so much.
Thank you.
Couple of questions. Could you just tell us, because I wasn't able to sort of follow along this bit in the talk, with the cells, JE1, 2, and 3, which one was the 8P here?
It's the 3, yeah.
Okay, 3 was the here. I thought so. I just wanted to make sure I was following that correctly. So
And
when
by
you
the way,
get to like,
Eitan and Bina, correct me if I'm wrong, because I mean, they just recently reviewed the
okay.
the types to me, but I think I'm right. I think 1 and 2 are parents, and the 3 is the proband.
Okay, great.
Yes, I think we were trying to blind ourselves for some time, but we got unblinded.
Yeah, inevitable. So, and then I guess my other questions are about the stability of the 8P genetic anomaly. So whether or not, when you get to that 6-month stage and you do the single cell, are you able to assess at that point if the 8P anomaly is the same, or has it changed?
That's a great point. I haven't thought about that. We can definitely regenotype the cells on the 6-month-old organoids and see if they remain constant. But on the parents, they are, right. I mean, sorry, on the probands, they are. You still see the 8P deletion there, right?
Yeah, I think it'll be interesting in tissue culture, though, that, because it's a much more stressful environment than a whole human body. And so you need that, don't you? You need to establish whether or not it stays the same to know whether or not you're still looking at what you think you're looking at, right?
Yeah. We did the genotype on the iPSCs just to make sure that with passages, the iPSCs will not change, and that remains stable. But we haven't done in the brain organoids, yeah.
I understand from like just normal standard iPSCs, and I'm thinking of someone's work at the Stanley Center, that CSMD1 gene it can be critical in the stability of the actual IPS line. And considering most of the 8P heroes with the deletion only have 1 copy, I think that's going to be an interesting point to look at in the culture as they go. And then I think the other thing that occurred to me during your talk, which was absolutely brilliant, by the way, is the age difference as well of, parents versus 8P hero, because you've got, I'm going to guess maybe like typically like a 30-year gap there. And I know from growing cells that like whenever we got newborn cells, they grew so much better than when we got older, this is the skin cells I was growing usually, than when we got older donors. So, do you think that might also be something that needs to be factored into the experiments, or are you not overly concerned?
Yeah, so once you get the fibroblasts and you reprogram them, both the proband and the parents go back to stage 0, right? I mean, we reestablish their neurodevelopment. So most of the time, I mean, we don't bother about the age of the biopsy, since we're going to reprogram them and restart everybody together. So I don't think that matters in here. At least, I mean, I think that's what the field more or less believes so far.
I should know this because this was like what my thesis was on years ago, but it was many years ago and I've done too many things since then. But do we know when we could do the reprogramming, like whether or not the telomeres actually get reset, like all the way, like sort of completely lengthened to what they would have been at birth? Do we know that?
Yeah, believe it or not, but yes. Yeah. It's
incredible. Sorry, it's hard to believe because it's just so incredible.
That's why he got the Nobel Prize.
Absolutely. Thank you. Yeah, that's pretty much my question. Thank you so much.
But the question about basically you're saying, is there just further like somatic evolution? Maybe that's a strong word, but basically there's somatic changes to the genotype where 8P itself over the course of these organoids that you might end up with some kind of mosaicism or something changing, right? Is that kind of what you were getting at?
I wonder, yeah.
Yeah. No,
whether or not, they'll shift. They're just not as genetically stable. They maybe not, I think it's definitely worth looking at to, whenever you do that experiment to make sure, it was still the same genetic, 8P variant that you thought you had at the beginning or you started with at the beginning.
Right.
Great. No, these are great questions. I think we'll have a kind of a recap of these questions, and hopefully, Whitney, we can follow up here with who asked them, what were the answers. If we don't know the answer, maybe we can figure out a way to keep track of that question and figure out an answer later. Stefan, please, you're next. Oh, and you're on mute.
Okay, this shouldn't happen, 3 years after the pandemic started. Great questions, Faye. I crossed a bunch off my list. Great talk, Alysson. I had a quick follow-up question, because my lab, we're getting started with MEAs as well, and it played such a central role in how you set up, how you plan on interpreting the data, and and what relevance it may have, and what value it may have for testing drugs or gene candidates. So we're thinking actually of doing this in monolayer, because in monolayer, our cells, if we do the undirected differentiation, we do get electrophysiologically mature cells with synapses and astrocytes that are nurturing the synapses. Within 3 months, we start getting mature action potential. So about 12 weeks or so. Are the organoids hardier, or is there some other reason why you invest the extra time to make a 6-month organoid that becomes electrophysiologically mature?
Yeah, it's because of we are interested on the evolution of these networks. Once you have like the 2D or even directed differentiation, yeah, you get your network, but they will not evolve. They would stay the same for throughout, and that's useful. I mean, if you're doing screening, if you're trying to understand how the neurons are communicating, that might be enough. But we really want to go to a stage where we have these brain oscillations, because the ultimate goal for us is really to look at the oscillations in the patients and compare to the organoids. I think if we do that only in tissue culture, it's going to be harder to bridge this behavior aspect of the conditions. That's the main reason why we're pushing.
Okay, and then just a clarification question. So following up on Faye, so this trio, this is the INFDU-DEL family trio, or is it okay?
Yep.
Ethan is nodding. Okay, gotcha. Okay, great. Thank you. Oh, I have one more. Sorry.
Go ahead.
Sorry, another shop question. Sorry, I'm talking shop. Alysson, what about calcium imaging? Have you thought about that? Have you used it?
We sometimes, yeah, we do in parallel some calcium imaging. It's hard to do in 3D, but we are having like some better readouts now that we can do calcium in parallel. Yeah, I mean, definitely possible. Yeah.
Okay, thank you.
Excellent. Any other questions or comments? Got about 12 minutes here remaining if we wanted to use it. I guess one question I had, going back to the Rosen example. So is the idea here to set up a kind of repurposing, an in utero type of repurposing, where you would want to actually intervene in utero, or is that considered, would that just be sort of so ethically or otherwise challenging? I don't know, it doesn't seem scientifically challenging given the Sovaldi case, but is that kind of where this is going? And should that be the template that AP is thinking about, is if we could repurpose a mechanism or find a drug that hits a mechanism that would potentially, play a role in utero during development, then you'd want to dose as early as possible. Is that kind of where this is all going?
Not really for us, Ethan. I don't think that's the major goal, because that will require like a genetic screening in uterus before the treatment. Maybe we're gonna get there in the future, but right now our goal is really on treating the individuals who already have the condition. And you might say, well, I mean, but there is no migration in a more developed brain. But our feeling is that Rosen is doing more than just migration. It's actually helping the neurons to form new synapses as well. So migration was just one of the readouts, but I think there are more mature readouts that we can use Rosen for. And ultimately, the organoid model as a model is really capturing neurodevelopment and not so much a more adult brain. So these are questions that only upon a clinical trial that we're going to know if it's going to work or not.
Great points. Anyone else have any questions or comments? Bina, any thoughts from you? Otherwise, I think this, as we said, these are all available on replay. I think there's also a YouTube channel, I believe, where you can watch these as well. But now that we're gathered live, if we have any other, anything else we wanted to talk about, now is the moment.
No, I just wanted to say thanks to everyone. I think, you know, again, this is just the beginning phases of all of this. So lots more to come. And I think that as some of the other researchers also have more information and, you know, certain tools, and then we can really start putting our heads together. So, you know, it's appreciated. And if we can expand the scope to, I think, some other families, and I say that because I know how difficult that is, you know, it might be something that we just have to kind of plan for in the future. So again, so it's so great to have everyone here together. And I know there's quite a few scientists that weren't able to watch, but we'll send them the recording. So Alysson, thanks again.
And just so we have a sense, Alysson, what's coming next? So the single cell RNA-seq, I guess from those 1 and 2 month samples, are those sort of in progress? What can we sort of expect is going to happen over the next sort of few weeks? And then I know you had to restart some of the cultures as well. So that's another clock we have to wait on.
So we collected already up to 5 months. So we are actually ahead of the schedule. I mean, from the schedule that I put on slide 1, we are way ahead of schedule. And we're gonna send that batch already to RNA-seq because I think we can already extract some data. And then, I mean, the second batch will work not only as a validation, but if we want like the later time points, we can do it as well. But yeah, so the RNA-seq will be started soon. And then it's a matter of analyzing the data, making sure that we have all the cell types and looking for the differences in the cell type. So that's more on the bioinformatics side. On the experimental side, we also have some organoids frozen where we're going to stain them, cross-section them, stain for the major cell populations, and then based it on the bulk RNA-seq or single cell RNA-seq, we will know what are the potential defects and use that to validate as well. So I think we are, the next month or so, we're probably going to have like some preliminary data coming from the gene expression.
Very exciting. This is such important foundational work for the community, and will generate so many hypotheses. And yeah, hopefully another Rosen example a case study will emerge here. Well, I think with that, thanks everybody. If there's no other questions, we can bid each other adieu, and this will be available, as Bina said, on replay. Thank you so much, Dr. Muotri, for sharing all of these insights and data, early data with us. Everybody, I can speak for everybody here. Yeah, we're just very grateful to see data and to also have it presented so clearly.
Thank you
so much. Yeah, thank you guys. Bye-bye.